Hi class, so we're going to go through hematopoiesis in this video, okay, of all of the formed elements of blood, red blood cells, platelets, and all of the white blood cells, okay. All right, so we're gonna start with erythropoiesis, okay. So first, all of hematopoiesis occurs in red bone marrow, okay. So that's found at the ends of our long bones and the epiphyses of our long bones and in flat bones, okay. And these all start with hematopoietic stem cells or hemocytoblasts. So these are the stem cells that give rise to all of those formed elements of blood, okay. So let's start with erythropoiesis. Okay, erythro like erythrocytes. Poiesis is esis is formation. So formation of red blood cells. Okay. So this occurs during hypoxia. So when we're getting low oxygen to our tissues. So again, that can be due to if we've lost some blood. So if we've had a large cut and have lost a lot of blood. If we're in an area of low oxygen, like high altitude, so we're climbing a mountain, for example, if we have anemia, things like that would cause hypoxia, okay? And this stimulates erythropoiesis. So first, what components do we need? So what do we need from our diet, okay? So first thing we need is iron. Okay, iron. So this is from our foods. We can get these from leafy green vegetables, and meats, and other ways as well, or through iron supplements, vitamins. Okay, so we take that in, we ingest that. This is the stomach here, and then here is the duodenum of the small intestine. It's the first portion of the small intestine. Okay. So in the intestines, this iron is going to be reduced to iron 2+. 2+. Okay, so from 3+ to 2+. Now this can bind with ferritin. Okay, and then that will cause ferritin to bind with other ferritin molecules to form hemosiderin. Okay, so let's say our ferritin is just a circle, right? It's a protein, so it's a folded up protein, but we're just gonna draw as this little circle here. Okay, it's gonna bind to other ferritin molecules to become hemosiderin. Okay. Some ferritin will be released to the blood. So some of this ferritin is going to be released to the blood via a ferroportin transmembrane protein. Okay, so you have this transmembrane protein. Some of this ferritin here is going to be released into the blood from the small intestine. Okay. Then when it's in the blood, it can be converted back to iron 3+. Okay. Then it's going to bind to a transporter protein, transferrin. And then now it can be sent to red bone marrow, okay, where it's going to be needed for erythropoiesis. We also need intrinsic factor. This is made by cells in parietal cells in the stomach. Okay, so let's say this is the cell in the stomach. It's going to make intrinsic factor. intrinsic factor can then bind to B12, something else we get from our diet or supplements. Okay. And then B12 can get absorbed into the bloodstream in the ileum of the small intestine. Okay. Folic acid is something else we need from our diet. This can enter the blood through duodenal cells, so similar to iron. So it's going to enter, go to our duodenal cells, and then enter into the bloodstream. Okay. 3 other things we need: carbohydrates and fats for our heme group for hemoglobin, and amino acids for the globin, okay, for the protein portion. Okay, so these 3, carbs, fats, and amino acids, are what make hemoglobin, or what are needed to make hemoglobin. Okay, so those enter in the same way. So they enter into the stomach and they get absorbed through the intestines. Okay, so carbs, amino acids, and fats. Here too. We also need these. Okay. Okay. From our nephron. So again, this is the nephron is a structure found in the kidneys. Okay, this is what helps to filter blood. Okay, so here's our nephron. There's some capillaries in the nephron right here. Okay, so there's our nephron. From the nephron, there's a gene called erythropoietin, EPO. Okay, HIF, hypoxia inducible factor, will activate or transcribe the gene. Transcription. Okay. So hypoxia will cause this HIF, hypoxia inducible factor, to become activated. Okay. And now erythropoietin, this is the gene. Now erythropoietin can be secreted. Okay, and that's what we need to start our erythropoiesis. Okay. Okay. So now let's get into our stages of erythropoiesis. Okay, this whole process takes about 15 days. Okay, so our first stage, we start with our hematopoietic stem cell. Okay, so we have this stem cell here with its large nucleus. Okay, so here's our hematopoietic stem cell. This is going to transform into the myeloid stem cell line. Okay, this is going to go down I'll draw this here, myeloid stem cell line. Okay. Next, this is going to transform into a pro-erythroblast. Okay, there's our nucleus there. Okay, pro-erythroblast. This is our hematopoietic stem cell. Okay. So this is going to divide, our pro-erythroblast is going to divide many times, okay, and then transform into a basophilic erythroblast, which looks very similar. Okay, the nucleus takes up most of that space there. Okay, so this here is our basophilic. Okay, this is where ribosomes are going to be synthesized. Okay, so those stain blue. So let's actually stain this whole thing blue. Okay, oops, erased. Okay, we'll stain most of this blue. Okay, so our basophilic erythroblast. Okay. Next, we have our polychromatic erythroblast. Draw that here. Polychromatic, so polychrome, right? Multiple colored. So now large amounts of hemoglobin is going to be synthesized, which is red colored. Okay, so the cell now shows pink and blue areas. Okay, so let's draw some of our nucleus as blue here. It's going to be pink as well. It's not quite this color pink under the microscope. Oops. Yeah, it's not quite that color pink under the microscope, but we're going to draw it that way anyway. It keeps erasing on me. Let's just draw it as blue, and we'll draw some pink around it, okay? Okay, so you're going to have both of those colors there. Okay. So this is where we need our B12 and our folic acid for DNA to mature and condense. This is why also folic acid is very important when someone is pregnant, because that's it helps with DNA synthesis. Okay, B12 as well. Okay, iron is going to be used here as well, because again we need iron for hemoglobin synthesis, because iron is found in those heme groups. Those are what carry oxygen. Okay, and then carbs, fatty acid, or carbs, fats, and amino acids are going to be needed to synthesize hemoglobin as well, right? So for the heme group, we need carbs and fats. For the globin, we need amino acids, which are the building blocks of proteins. Okay, then we have our orthochromatic erythroblast. There's our nucleus there. So mostly hemoglobin here, so it's going to appear more of like a red or a pink color. Okay. This is where most organelles are ejected and the nucleus is starting to degrade. So you see I drew it a lot smaller here. Okay, so nucleus is starting to degrade. Okay, so it's getting smaller. And then it is pushed out. So the second portion of this is another orthochromatic erythroblast. It's just showing like the late stage here. Nucleus is now ejected. Okay, it's pushed out. And this is what's going to cause that concave shape to the red blood cell. Okay. And then we have our reticulocyte. Now it doesn't have a nucleus, right? It was pushed out from the orthochromatic erythroblast. And no mitochondria, right? So those organelles were ejected, and we don't want red blood cells to have mitochondria and use all of the oxygen that they're transporting. And then finally we have our mature erythrocyte. And I'm drawing this to show it's concave in this center part here, but it does not have a nucleus, so that circle inside is not a nucleus. It's just trying to show, I'm not a great drawer, but it's just trying to show its concave shape there. Okay. So now ribosomes are going to degrade, and we have our mature red blood cell. Okay, so that is erythropoiesis. Okay, now let's get into leukopoiesis. So leukopoiesis, this is formation of white blood cells. So remember, leuc means white. Okay. So we start still with our hemocytoblast. And this can go into the myeloid stem cell line or the lymphoid. It's only going to go into the lymphoid stem cell line when we need lymphocytes, B and T cells. Myeloid stem cell line is going to make all of our granulocytes and monocytes, which are considered agranulocytes, but again, they have granules. They just don't show up in staining. Okay. All right. So let's first go into granulocyte production. So we're going to start with our, use a different color here. We're going to start with our myeloblasts. Okay, these arise from our myeloid stem cell line. And then this is going to have 3 lineages as well. So basophilic for our basophil cells. Eosinophilic for eosinophils. And neutrophilic for our neutrophils. Okay, so from our myoblasts, let's just go here, we get our promyelocytes. Remember, every time you see pro, it's kind of like pre, okay? So next we would have our myelocytes. So promyelocytes, these are where lysosomes are accumulated. Then our myelocytes, this is where our granules are accumulated. Okay, so here, basophilic will show blue for basic. Eosinophilic will show red for acidic. And neutrophilic, both. Okay, blue and red, so it'll be like a pinkish color. The nucleus also starts to bend here. Okay, so we have a normal nucleus. It's going to start to bend. Okay, not quite a full bend yet. Okay. And then we have our band cells. Now the nuclei form, so from our slight bend to a full bend. Okay. So now it's like a fully curved arc. Okay. So now we have our mature granulocyte, so either our basophil, eosinophil, or neutrophil. Okay. And the nuclei still needs to become segmented before it's released into the blood. Okay, so our basophil, we'll draw the whole thing in blue. Our basophil is going to have a nucleus that looks kind of S-shaped. So here's our basophil. Okay. So it's going to be more like an S shape. So it kind of looks like this. Okay, something like that. And it's going to look like filled in. Okay. When we look at these under the microscope, we'll see these in, we'll see it in lab under the microscope, but there's going to be like these large granules everywhere. So it's going to kind of cover that nucleus, okay? So it's going to be kind of difficult to see that nucleus under the microscope, okay? But just so you know what it looks like. It looks like an S like that. Okay. Next, we have our eosinophils. Okay, so these ones show up like red, so I'm just going to draw the whole thing in red. Eosinophil. Okay. It has a bilobed nucleus. So it kind of looks like aviator glasses. Something like that. Make this easier. Okay, so kind of like aviator glasses there. So that's what the nucleus is going to look like there. And there will be granules throughout here as well. Okay, because it's a granulocyte. Okay. And then we have our one that looks like has red and blue, so it looks more of like a pinkish color. It looks more kind of like a light purple. I'll just draw it as purple. Okay, so this is our neutrophil. Okay, our neutrophil. Okay, its nucleus forms 3 to 5 lobes. Okay, so it's going to be something like this, like that. Okay, so that's what our neutrophil is going to look like. Okay. All right, so that was all of our granulocytes. Okay, now let's get into thrombopoiesis. Thrombocytes are platelets, poiesis, formation. So formation of platelets. This one's very simple. We have our hematopoietic stem cell. So again, our hematopoietic stem cell with its nucleus. Just kind of color it in a little bit here. Hematopoietic stem cell. It's going to go down the myeloid stem cell line. Remember, lymphoid is only for lymphocytes. Okay, from a megakaryoblast. Okay, and this is a very large cell with a nucleus. That's going to become a megakaryocyte, which is an even larger cell. Okay, it's going to have its large nucleus. Okay. It's going to grow even larger. There's our nucleus there. And pieces of it start to kind of break off. Okay, and they break off into these small little platelets. Okay, that's it for thrombopoiesis. Pretty straightforward and simple. Okay. Okay, now we have our agranulocyte production of monocytes. Monocytes. These are derived from the myeloid line. Remember, they have granules, but they're not seen under staining, so they're considered agranulocytes. So this one's pretty straightforward as well. We start from a monoblast cell, go to a promonocyte, then to a monocyte. Okay, then it could monocyte. Then it can enter the bloodstream. And then finally, our lymphocytes. Let's draw a little line here. I'll do black. Lymphocytes. Our hemocytoblast goes through the lymphoid stem cell line. So we still start with our hemocytoblast. Lymphoid stem cell line. Okay. So from a hemocytoblast to a prolymphocyte, and then remember pro is like pre. So then we get our lymphocyte. Then B lymphocyte. And T lymphocyte. Okay, so the T lymphocyte is still immature in the red blood cell. It goes to the thymus to become mature. B lymphocytes will mature within the red bone marrow. Okay, and these lymphocytes can live years. Okay, so when we make antibodies towards something, right? So when we have like chickenpox, for example, you only get that once because you make the antibodies for it. Then anytime you are in contact with that virus again, those antibodies will come and fight them off. Okay.